Hello, everyone. Welcome to today's live broadcast, Magnetic 3D Bioprinting of Cells, Overcoming Imaging Obstacles in Spheroids. I'm Christina Jewell of LabRoots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots, N3D, Greiner Bio 1, and MD Anderson Cancer Center, and sponsored by GE Healthcare. GE Healthcare's cutting-edge imaging technologies continue to provide amazing depth and breadth of investigation. From extensive data analysis to super-resolution cellular imaging, GE Healthcare allows you to analyze interaction as they happen and gain deeper insights into complex cellular mechanisms. For more information, please visit www.gelifesciences.com/cell imaging. We have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of your presentation window. We will try to answer as many questions as we can. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you have any technical problems viewing or hearing this presentation, please click on the support button at the top right of your presentation window, or submit your problem through the green Q&A button in the lower left. This webinar has been approved for continuing educational credits. Please click on the CE button at the bottom left corner and follow the process to receive your credits. Our speaker today is Dr. Glauco R. Souza. Dr. Glauco R. Souza is the president, CSO, and co-founder of Nano 3D Biosciences Incorporated, N3D. Dr. Souza is also adjunct assistant professor at University of Texas Health Science Center at Houston and is one of the co-inventors of N3D's 3D cell culturing and magnetic 3D bioprinting technology. These groundbreaking technologies have been reported in various high-impact scientific journals, including Nature Nanotechnology and Nature Reviews Cancer. For Dr. Souza's complete bio, please see labroots.com. I will now hand over to Dr. Souza for his presentation. Dr. Souza?
Thank you, Christy. Quick note, I'll be the only one speaking, but I'll be showing some remarkable results from Dr. Jeff Bartolomeo's from MD Anderson Cancer Center here in Houston. Quick, who is N3D? We are a spin-off from Rice University and MD Anderson Cancer Center. We're located right at the heart of Houston Medical Center, and we are a close partner with Greiner Bio 1. Greiner is our partner that commercializes our systems. Of course, when I give a presentation, even though I'm preaching to the choir, I have to give a bit of motivation of why we should do 3D cell culturing. And it's not only going to be this slide, the motivation. I hope that the results I'll be showing is emphasize the importance and the value of doing 3D cell culturing. And this slide is a simplified version of how the drug discovery process takes place. And we always hear that to develop a drug, it takes so long, and it's always very expensive. And part of the reason for that is because the very first step that we do in the drug discovery process is using cells cultured in monolayer, in 2D. And today there's enough results to show that 2D cell culture is a very bad predictor of in vivo biology. And the next step is using animals. And animals are usually very expensive, and they're not humans. So ideally, we would like to have to do experiments in 3D using human cells, and that's what we're doing with 3D cell culture. We're bridging the gap between in vitro biology to human biology. But we're not only bridging the gap between in vitro and humans, but we're also trying to meet the challenge of doing 3D cell culturing using microscopy tools, high throughput, and high content imaging, which is not often an easy task when dealing with 3D cell cultures. So in this webinar, I'm going to be showing you magnetic 3D bioprinting, and I'm going to show you that we can easily do 3D cultures. We can co-culture different cell types. We use flat bottom with high optical quality surfaces. I'm going to show you high throughput, 96, 384, and 1536 well systems. We'll see that magnetic 3D bioprinting is compatible with high content analysis. It's compatible with confocal and wide field microscopy. It's compatible with liquid handling and automation. And you can use time-dependent imaging, which is key. So we like to call ourselves the system that is 3D on a 2D workflow. And one thing that is important to highlight is that magnetic 3D bioprinting, because of many of the practical aspects that we enable, take full advantage of all the amazing tools you have with InCell Analyzer 6000. And all the images that you're going to see in this webinar are not collected with InCell 6000. However, I expect if they were collected with InCell 6000, they would be of the same quality or better. So how do we do what we call magnetic 3D cell culturing that we separate into 3D bioprinting and levitation? We have what we call the nanoshuttle. The nanoshuttle is a magnetic nanoparticle assembly made of gold and nanoparticles, iron oxide nanoparticles, and polylysine. And the polylysine basically provides the attachment of the nanoparticles to the cells electrostatically. It's very simple. You don't require any special media. You just add the nanoshuttle to your cells. They stick to the cell membrane. We can use cells in monolayer or in suspension. And then you detach your cells using whatever method you use in your laboratory. And you can use what we call magnetic levitation, where you put a magnetic field on top. The cells literally levitate off the bottom of the surface and aggregate in suspension. And this we have in single, 6, and 24 well format. And we have the bioprinting, which is on the bottom right, which is we have in 6, 24, and high throughput, 96, 384, and we're about to launch the 1536. And again, on the bioprinting, the magnetic field is outside of the well, but on the bottom. The key for our technique, it's not the magnetic field or the nanoparticles. It's cell-cell interaction. Within the very few minutes of our culture, as you can see in this quick movie, the cells come together and we promote cell-cell interaction. And this shows the importance of the cell-cell interaction. So what you see here, we took a human primary fibroblast. We cultured for 6 hours, 12 hours. We fixed the cells, and we stained for laminin. The red is laminin. And the top row is non-permeabilized, and the bottom is the permeabilized. So since the top row is the non-permeabilized, you can see the red staining on the 6 hours and 12 hours, which shows the deposition of extracellular matrix very rapidly. And this is endogenous. The cells are making their own extracellular matrix. And we've published this results in Nature Protocols and similar results on tissue engineering. One of the first results we have, which is key, addresses the question that people have: why should we use 3D cell culture? Does 3D cell culture really look like when cells are grown in vivo? So this was one of the early experiments we did, where we took L929 human glioblastoma, did a mouse xenograft. We cultured the cells magnetically as well, and we did 2D. So the left is the paraffin embedding result of the xenograft that took 30 days to grow. We did our culture in the middle. It shows the culture, the cells culture with our system for 48 hours. And on the right is the two-dimensional. So the middle row, you can see, is the Encagerian staining, and you don't have to be an expert to realize that 3D culture by magnetic bioprinting is very similar to the in vivo, And that's a key result that actually got us to probably to publish in Nature Nanotech. So we have generated several models. We have, of course, the spheroid models, and on the bottom left here, you have fibroblasts cultured for 12 hours. So when we bring the cells together, they can assemble very fast into spheroids. We have done co-culture models. On the top right, we have glioblastoma, which is GFP label, co-culture with normal human astrocyte, M-cherry labeled, which we magnetically cultured them separate, and then we magnetically brought them together, and we could observe the invasion of the glioblastoma into the normal astrocyte. And on the bottom right is a co-culture of pre-adipocyte stem cells, which were differentiating to adipocytes and endothelial cells. And I'll give you more detail on these results in a few slides. So this is actually a very cool result from a collaboration with the University of Houston, Dr. Birla, and where we took a rat's heart, we digested the entire rat's heart, magnetized and reassembled, and after 24 hours, the culture started beating. Please play movie. So, and this is a key result because when we publish our results, people always ask for function, and what we just showed is it's phenotypical, but it is function of the cells we cultured. And I always show this slide for a few different reasons. This is a new technology, and in our world, in the scientific world, we validate a technology by publishing, and we've placed a huge effort in publishing in very good journals, high-impact journals, and as you can see, we've been fairly successful. But also, I showed this because we use nanoparticles. And as you're probably asking, wanting to ask, what do the nanoparticles do to the cells? And whenever we try to publish in these journals, that's the first question they ask. So we have published the answers. And in one example I like to give you to realize that the nanoparticles are in very small concentrations and that has little effect on the cells. The analogy is you have a headache, you take an aspirin, the headache goes away. If you take 10 aspirins, there's a good chance you're going to get an ulcer. It's the same idea here. We use a very small amount of nanoparticles. The cells are very buoyant. So we just need a few nanoparticles to stick to the cells. We can apply the magnetic field, and they come together. So the nanoparticles are very much bioinert to the cells. And we have published this. And if you have any questions, you can contact us. We can give you a white paper with the summary of all the results we have. So this is one of studies that we did with collaboration with Dr. Misha Coloni here at UT Health Science Center in Houston, where they wanted to do a co-culture of pre-adipocyte stem cells and endothelial cells in a differentiated pre-adipocyte stem cells to generate a more in vivo-like model, in vitro model of adipose tissue. So what was interesting from this study is when they first approached us, they asked which ratio of cells, what is the ideal ratio to achieve a tissue that looks like fat in vivo. And to be honest, I didn't know. I told them to use 50-50, 50 of pre-adipocyte, 50 on the endothelial cells, and I tell you, it didn't work. The cells didn't show the morphology we wanted. But because we can magnetically control the cells coming together, so we could basically titrate or vary the ratios of the two cell types. And for us to achieve this adiposphere, where red here is pre-lipin, which is a marker for adipocyte, the green is GFP labeled endothelial cells, we had to use 95% of pre-adipocyte stem cells, 5% of the endothelial cells to achieve such a structure like this. And this image here was the cover of the Tissue Engineering that we published these results. So, and again, the morphology is very similar of the morphology of the in vivo tissue. It's very similar to the 3D assembled tissue. Now on the right here, WAT is wide adipose tissue. You can see you have these multinuclei regions with what we call lipid pockets, and it's very similar to the 3D levitated culture, which is very different from the 2D, the stem cells differentiated in 2D. In 2D, the cells have these blisters, as you can see here. This is oil red on the left, where you don't see that in vivo. So this was a great result. Again, we're showing that the cells culture with our system is much more look much more like in vivo. But from the first results, this one where we had to use the 95% to 5%, 95% pre-adipocyte to 5% endothelial cells, made us raise the hypothesis of what about if we bring together the ratio of cells that is the correct one for them to assemble into in vivo-like tissue. And for us to do that, we figured we need to take in vivo tissue. So we took the whole wide adipose tissue of the rat, we removed the adipocytes, we magnetized the stroma vascular fraction cells, all the cells, and we brought the cells together. And after 2 weeks, on the bottom left here, the tissue organized, clearly organized. And what you see here in green where the arrows are pointing to, that's CD31 staining. So it seems like the tissue organized, what it looks like initiation of a blood vessel formation. And on the right, the green is decorin, which is the stroma cells, which seems to be in the right placement in the culture. So the bottom line is if you have the right ratio of cells, they will organize and do a lot of the work for us. Now I'm going to shift gears into magnetic 3D bioprinting, which is a very simple technique. Bioprinting, people usually think that we have some very fancy instrumentation. We can couple this to automation, liquid handling, sure, but this is a kit. And basically, to do the magnetic bioprint of spheroids or rings, you need a magnet drive. As you see here on this plate, you see this plate with magnet pins, and then you just placed your micro-well plate on top of these magnet pins before dispensing the cells. The surface of this micro-well plate does need to be cell repellent. So we use the Griner cell repellent surfaces. So as you remember from the earlier slide, we magnetize your cells, then you're going to transfer to the micro-well plate. And when you transfer the cells to the micro-well plate, they go directly to the magnet, as you can see in this bottom image here. So after you add your cells, you wait just 15 minutes on the magnet, and you remove your magnet. And then the cells will form the spheroid. So whenever you place your tissue plates after dispensing your cells in the incubator, you don't need to have the magnet anymore. You're going to put the plate back on the magnet if you want to manipulate, if you want to change media, do immunohistochemistry. Then you're going to place on the magnet so you don't lose your spheroids. So this is a picture of a 384 well plate. You can see all the spheroids centered in the middle of the well, which I'll show you in a little bit. This is a very helpful feature, especially when you're doing high throughput imaging or high content image as well. And I'm going to change gears here for a few slides and show some really exciting results from Dr. Jeff Bartolomeo's laboratory at MD Anderson. And he's using our system to culture cells from patient-derived xenografts. And I'll show some results, our results using biopsy samples as well. So just to give a little bit of history on this project. First, Dr. Bartolomeo's wanted to use no matrix models. He didn't want to use matrigel or hydrogel to generate the 3D cultures from using cells from PDX. And he didn't choose our system at first. He chose the Cyvex plates, which are basically are micro plates with micro patterns on the bottom that induce the cells to form 3D culture. So he took his PDX tumor, he broke it apart, he put it on the Cyvex plates, and this was the result he got. He got the cells all dispersed in the well, and they didn't really come together. He did see some of these structures, the cells to form some 3D structures, and he labeled them, and he found out that those were not cancer cells. Those are stem cells, actually. So long story short, it didn't really work. So when I talked to him, and we saw that by adding the cells to the pattern surfaces, the cells just stay dispersed. They don't come together. So my suggestion was, let's magnetize your cells and use the magnet to make sure the cells stay together. And that's what we did. So, but one detail that is important for me to point out, most of the general protocol calls to leave the magnet on for only 15 minutes. For in case of PDX cells, it is important to leave the magnet on at least overnight. And the reason is that the tissue processed from the PDX model, you're mincing, you're breaking the tissue apart, keeping the magnetic force to bring the cells together will show that it's really key to keep integrity of the samples. But the question still, so he was able to take the PDX tumor, break it apart, and he was able to generate a viable culture with our system. But the question still remained, how does it compare to the animal tumor? Right? So what we did, and this is a diagram that shows the experiment that was performed, that is being performed in his lab. We took a PDX from breast cancer PDX. Here's the tumor right on the top in the middle. And we cut the PDX tumor in half. So as you go to the left here, the half we paraffin embedded, and we characterized using immunohistochemistry. On the right, the other half, we basically broke the tissue apart, magnetized the cells, placed the magnet, and magnetically assembled the tissue. And we did the same thing. We paraffin embedded, and you did immunohistochemistry. Actually, with our system, because we had several wells, we can do actually dose-dependent studies. So this is the outcome. On the left, you have the tumor xenograft. On the right, you have the bioprinted culture. As you can see, the top is icadierin, viamentin human and mouse, Ki67, PISMAD. You can see that the cultures from bioprinted cultures are remarkably similar to the tumor xenograft. And this is the type of data people are looking for to give them confidence that moving to 3D, you can have more in vivo-like culture. So again, this is, we're comparing tumor xenograft with two separate cultures bioprinted from the same tumor. So it's remarkable that the similarity between the two. And this work should be published pretty soon. So, and of course, when you're doing this work, you always have to check, are the cells viable and for how long? So here's just showing that he bioprinted the cells, and the viability at day 0, at day 5, it did increase. It's not a huge doubling of the cells, but if you have handled cells from xenograft tumors, a lot of these cells don't grow very well at all in vitro. So the fact that we could maintain viability for 5 days and actually the cells grow is very relevant. And now he's actually taking a step further. He's actually taking the same tumor from the patient, putting different animals and comparing if there is difference in dose response between the cells from the different animals. And this may address a few different things. One is the heterogeneity of the original tumor, and also if there are differences between tumors grown in different mice. So here is you have tumor A, mouse with tumor A, mouse A basically, and tumor from mouse A, tumor from mouse B, and you can clearly see you have drug concentration and you have viability, which is chemiluminescence. You can see that the dose response between the two tumors is different. And he did for carboplatin, AZD6244, and he also did erolinitinib, and my apologies for not pronouncing this correctly. And there was also a difference in the dose response from these tumors. This will be published very soon, and he's actually doing the same type of dose-dependent studies in the mouse to compare. So this is a key result that should really push us in the right direction. Of course, we've done the same with prostate cancer xenograft. We can take 1 gram of tumor. We have a method to disperse the cells. Basically, 1 gram of tumors, we can extract 100 million cells, and this gives thousands of wells, printing cells at 10,000 per well. And but this is on the bottom right here is the dose response for 4 different compounds using a single 384 well. Very good dose response curves, and the beauty of our system is you can put it back on the magnet and do the immunohistochemistry, which is very hard to do with other 3D cell culture, and that's what you see here on the top right. And we actually took a step further, where we took actually a biopsy sample. This was in collaboration with Dr. Bob Amato from ETU Health Science Center. Here in Houston, we're close collaborators, and we took a bone biopsy. We had enough sample to disperse, magnetize, and reassemble and get a dose dependence for doxorubicin. And it's a very good dose dependence curve. And again, we fix the cells, we put it back on the magnet, and we can stain for EpChem, which is a marker for these cells, using a whole mount procedure, just like you do in the 2G. And these results, I do believe, are key for us to move into the precision medicine area, where you can take a small piece of a tumor biopsy and get a lot of results with or without doing a PDX. So this is what the kit basically looks like. You have 2 magnet, what we call magnet drives. You have a small magnet and a larger magnet. The small magnet is what we call the dot printing drive, which we use to assemble your cells for 15 minutes. And we have the larger magnets. If you want to do your washes, you want to change media, you recommend it to put on the larger magnet just to make sure it's you don't lose your structures. And the cell repellent surface plates from Griner. So now I'm going to show you more of the traditional 3D cell culture type of results. But again, these are spheroids printed from fibroblasts, and this is just 12 hours. And you can see very reproducible in size. And it's important for me to point that this was done with InCell 6000, and it's 4 times magnification only. But even though it's 4 times magnification only, the resolution you get from these spheroids is remarkable. So, and again, this was done without needing to look for the structures in the well, because they're all centered magnetically right in the middle before imaging. And one thing that people often ask is, do you have the magnet when you image? No. The magnet is just to assemble the cells quick or to center them. That's all. So, and of course, InCell 6000 provides all the software to do the detailed analysis of your spheroids. You can get area, form factor, average diameter if you're imaging several spheroids, perimeter of the spheroids, and it's fairly straightforward to use. And of course, we're characterizing our spheroids. Here is our HepG2 liver cells printed in 384. You have on the right 600 cells, all the way to 20,000 cells. And you can see that number of cells versus diameter on the top left plot, and the chemiluminescence signal on the bottom left. You can see that it's very reproducible in size and very linear signal as far as the viability signal using cell-titer glow. And this data is published on Promega's PubHub as a collaboration with Promega. One area that is 3D cell culture is very relevant is with testing for toxicity testing using liver cells. These are results using iPS-derived hepatocytes, where we show the metabolic activity of the 3D cultures is significantly higher than with 2D. And here we just use rifampicin to induce the metabolic activity of the cells, and we use the CYP assay from Promega to detect the metabolic activity. And as you can see, the red here is increased metabolic activity due to the CYP activity. Of course, we also have cardiomyocytes is another cell type that we frequently get asked if we can use our system to print cardiomyocytes, and this is using iPS-derived cardiomyocytes from CDI, and we printed 1,000, 5,000, 10,000 cells with the 384 system, and I'll show you the movie of showing the beating of the cells. Please play movie. So again, we printed the cells here, and then overnight they start beating, and they sustain the beating for 7 days, and it works really well. So again, since what we do is very relevant to high throughput and high content imaging, it's very challenging when you generate your 3D cultures. And a lot of the times when you're generating 3D cultures in matrix-free systems, the spheroids or the 3D structures tend to float to the side. As you can see here, the red arrow on the left image point out the wells that there's no spheroids in there, or they're stuck to the side. And so that's data loss. And as you can see, you lost probably a quarter of the cells here. And this was in collaboration with, these results are in collaboration with the Scripps Research Institute in Florida, the group of Dr. Tim Spice. And when you want to do your imaging, if you place the plate back on the magnet for 30 seconds, you recover 100% of your spheroids right in the middle of the plate, which make it much easier for imaging, and you don't have to scan, waste time when imaging yourselves. One aspect which you already mentioned before, but now is in the context of high throughput, is co-culture different cell types and controlling the ratio of the different cell types. And here we took A549 lung adenocarcinoma mixed with primary fibroblast or tracheal smooth muscle. We mixed them together and we let them reorganize and interact for 48 hours. And when you come back and look at them, the green is the HFP on the top row, the bottom is the smooth muscle, and you can clearly see that adenocarcinoma, the cancer cells, like to stay on the outside, as shown in red. But the important aspect here, we can control this the ratio, and we can make sure the cells are coming together. So this is quite interesting. And we did the same now with CALU3, lung adenocarcinoma and fibroblast. And interestingly, this time the fibroblast prefer to organize on the outside and the Calu3 in the inside. As you can see here, the green is the fibroblast and the red is the Calu3. These are 4 different cultures, so you can see that this process is very reproducible. And again, it's just the cells organized. This was taken with InCell 6000, and I'll show you the confocal movie that actually shows you through the different Z-stacks that the tissue, the fibroblasts are localized on the outside, which are the green cells. And just to remind you, this is only 12 hours of culture, so this is really fast. Please play movie. As you can see, the green is mostly on the outside, and this structure is still formed a very spherical shape. As you can see from the previous image, they're not perfect spheres yet, but they definitely have the circular aspect. And to tell you the truth, not all cells like to make a spheroid, but this did generate spheroids at a later time point. And this is the characterization of those cultures. So on the left, you have your fibroblast-only culture. The middle, you have the co-culture of the fibroblast and Calu3. On the right, the Calu3. And the InCell has all the capabilities to measure the InCell software, the area of the spheroids, the spheroid form factor, and the perimeter of the spheroids. What is important here is that, as you can see from each row you see from on the graphs on the right is a different spheroid, and they are very reproducible from spheroid to spheroid. And this is very important because it demonstrates how precise the printing process is. This is, again, this results from Dr. Jeff Bartolomew's lab, and here he actually is he's culturing 3 different cell types. On the left, you have RFP-labeled astrocytes, the first 3 columns. On the middle, they are breast cancer from a brain metastasis site, and then you have breast cancer from a lung metastasis site. And what is interesting here, in each column you see here, you have different cell numbers. You have 10,000 cells, 20,000 cells, and 30,000 cells on the 3rd row. And the breast cancer cells are GFP labeled, as you see in the middle stack of cells. In the bottom, you see the RFP labeled cells, which are the astrocytes. But what is interesting is that when you bioprint the cells into a spheroid, the brain metastasis has very different morphology from the lung metastasis. The lung metastasis has more of a loose structure, and of course, the astrocytes generate a very tight structure. And again, this is overnight, and we can collect these images without having to scan through the well. All these pictures were taken. It's 4-time magnification, but all the spheroids were centered when imaging. So they took a step further. They actually mixed they did the co-culture of the astrocyte with the breast cancer from the brain metastasis and the astrocyte with the breast cancer from the lung metastasis, as you can see here. And the morphology of the 2 co-cultures is different as well. As we saw with the single cells, the lung assembly is more loose. But what is interesting or relevant is that the cancer chose to organize outside of the fibroblast. As you can see from the GFP label in the middle, is the cells, the cancer cells are localized on the outside, and the astrocytes is localized in the middle. And again, this was all images taken with InCell 6000. Now we're going to take a step further. We're pushing from 384, all the results you saw until now were 384. Now we're moving into 1536, which is, on the right is the image of the prototype for what we call the magnet drive. And the zoomed in region showing the little magnets, that's a 9.6 well. But what is important, and it's a recurring theme on this presentation, is as soon as you put your cells in the well, when they're sitting on the magnet, these cells come together within 15 minutes. And as you can see, you have on the left here, you have an image of colon cancer and the lung adenocarcinoma A549, and you have the left is bioprinted, and the right is only when you throw when the cells are sitting in the well without the magnet. You can see that clearly that the cancer cells are all assembled all together after just 15 minutes of bioprinting. And again, the images have no magnet. The plates were on the magnet for 15 minutes on. And we're developing the system with Greiner Bio 1. And we should have, we're hoping to have it available in the next couple of months. And this image is from the 1536 plate. And again, this is 10 times magnification. All the 3D cultures were centered in the middle of the well. We did not have to scan the wells to capture all this image. So it's the same field of view, basically. And you have 1,000 cells, 500 cells, and 250 cells on the different rows, as you can see from the label here. So it's a very good, very efficient way of generating 3D cultures using 1536. And again, this will be is a key technique for the high throughput, because I don't think you're going to be able to generate a system using curved surfaces, which you have for 384 and 96 well, especially a system that is compatible with imaging. We know 384 is all the curved surfaces are challenging to image, especially when you want a high resolution and confocal, which is absolutely, if you're doing high content and high throughput using the E-cell, you're going to need it. So I just want to point out the surfaces that we use are from Greiner Bio 1. They have a wide range of different plates, flat bottom and high optical quality. Now I'm going to shift a little bit of gears to what I usually call this the academic part of my presentation, because it's a new way of looking at cells. So we know by using magnetic field, you can bring the cells together very fast. And by doing that, when we start observing what the cells do once we bring them together, because remember, we bring the cells together for 15 minutes, then we remove it from the magnet. And it's interesting that some cells, they tend to contract, as you see to the left. Some cells tend to expand within this very few minutes. And this, we're not taking into account cell duplication, cell growth, really cell-cell interaction. So because we can bring the cells together very fast, our question was, can we use this process of reassembly? It's almost a homeostatic process to look at toxicity, a drug effect, at cell function, at viability. And the key for that is really using real-time imaging. So, and we have two approaches. We have the ring, we have the dot, as most of the data you've seen is using what we call the dot, which you basically print a spheroid and you watch the cells contract and expanding. But we also have a ring. You can print a ring. As you can see on the left here, basically is a drive where you have a ring magnet, and you can generate a ring just like a sort of similar to a wound healing assay. And so we can generate that very fast, and you can watch the cells to close the ring. And this is sort of a comparison of the traditional and what we call rapid phenotypical that we're starting to develop and take a full capability of the 3D bioprinting, which is being able to observe cells early on the tissue assembly. So on the top, you have the traditional. You make, as you see on the left here, you make a spheroid and you watch it grow. And it usually takes 10 days to do those assays or more. So it's very time consuming. On the bottom, you have the rapid phenotypical. You assemble the tissue for 15 minutes, and within hours, you can get information from your system. And we have 3 very good publications on this describing the system. And again, there is value in observing, especially if you're probing mobility of cells, cell migration, cell-cell interaction, cell-extracellular matrix interaction. There's definitely value in using this approach. We actually published with AstraZeneca vasoactivity assay, which we print a vascular smooth muscle, and we can follow the dilation contraction. And to the audience that does cell culture in general, we all know that time is not a friend of whoever's working with cells. We know that it's always challenging long-term cell growth because cells can change, cells can die. So if you can get early on results in hours in a label-free mode, I do believe there's tremendous value. And this is a result that we did, that we demonstrated. We did the ring assay. We basically took GFP fluorescence cell tracker labeled fibroblast. We used the ring, we print into a ring, and we used the InCell 6000 to follow the process of cell contraction. Please play movie. And this is my apologies. What I meant to say is that we're going to replay the movie a few times. So what we're really looking at is the contraction of the ring. So, and that is, you can quantify that, and that has, as you see on this publication, there's a lot of good information you can get from this essay. And again, it's new. The reason we can do it is because we can print the cells rapidly. And again, we're trying to bridge the gap here, not only from in vitro to human biology, but also we're trying to meet the challenge of integrating 3D cell culturing with microscopy, with high throughput and high content. And just to go over a few things we learned, I hope we learned today, is we're doing 3D cell culturing high throughput with flat bottom, high optic quality surfaces in 96, 384, 1536 well format. We can do high content analysis, and again, confocal and wild field microscopy. Again, because the cultures are magnetized, we can do liquid handling and use liquid handling automation by placing the magnet on the bottom, just like you do with 2D. As I showed in the last piece of data, time-dependent imaging is key, and there is tremendous value. And we are a 3D on a 2D workflow because the magnetized cultures really allows us to use automation and take full advantage of imagers like the eCell Analyzer 6000 from GE. Of course, I have to thank our corporate partners, Griner Bio1, of course, GE, ATCC for providing us with the cells, MD Anderson, and UT Health Science Center. Of course, we're a small company. This work is never done alone, as you all know. We have great collaborators. We have funding from NIH, from NSF, from cases from the center of the cases is the Center for Advancement of Science in Space. We actually have a grant from them, and our system is going to a space station in December. And what people usually ask me is, why are you taking a 3D system to space when cells in space are supposed to go in 3D, right? For two reasons. One is that cells are, although they grow in 3D in space, they're very hard to manipulate. So exchanging media is imaging. It's not simple at all. So basically, we're sending magnetized cells and using the magnets to hold the cultures in space, so you can change media, you can add a compound, and facilitate the work of the astronauts. And we're actually going to go the reverse. We are actually going to use our system to print cells in 2D as the control, the 2D control that we haven't had in space. So by sending the magnetized cells, we can use the magnetic field to basically smash the cells to a membrane and force them to go in 2D to actually have a control between cells going 3D and 2D in space that we don't have. So this is it's pretty cool. And of course, I have to thank Jeff Bartholomeus, GE Healthcare, Paula Feinberg, Zedek has been great help in getting these images. And I'm open for questions. Thank you very much.
Thank you, Dr. Souza, for that great presentation. While we get ready to answer some of your questions, we have two polling questions that we would like you to answer. They will be showing up on your screen now. Please answer each question so we know how best to follow up with you. Thanks. Now, during the question and answer session, I would like to remind our audience how to submit questions. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. And we'll try to answer as many questions as we can. So our first question is, Dr. Souza, are preparation conditions very cell type specific? In other words, would I expect a lot of assay development time if I choose to use a new cell line?
My answer is different cell types require different conditions, period. So if you have an optimizer system for, let's say, for monolayer culture, which people usually have before using 3D, you should use the same conditions. So the answer is no. Transitioning from 2D to 3D, you shouldn't really you should use the same conditions, basically.
Next question. Have you done a comparison of cell viability between the magnetized cells and 2D cultured cells over a lengthy period of time?
Yes, we have, and we have published in different journals, and people always ask that question. We always show nanoparticles and non-nanoparticles, and we've seen no difference. And on In3D's website, we actually have a white paper that basically compile all the published results and in-house results that we have showing the biocompatibility of our nanoparticles.
Next question. For the pilot drug screen, were drugs added after assembly of the spheroids or before? And could combining these approaches be a new way to study drug delivery efficacy?
My answer is, depending on the question you have, you may want to add your drug either before, during the assembly, or after the assembly. Because we're very flexible. That's the beauty of our system. Because the assembly of the cells is fast, you can actually get a lot of information by adding your compound while they assemble or after it's assembled. But you may see a difference on the results, yes, which makes sense, but you can control that very easily.
Next question. Is the nano shuttle cytotoxic? And can I aspirate the media out of wells without losing the culture?
Yes, as I answered the first question, yes, the nanoshuttles are not cytotoxic, and we have a lot of published data to show that. Can you aspirate the media without losing your cultures? Yes, and that's one of the great benefits of our system, because you can put these cells back on the magnet, and you can do, basically can exchange media, you can refresh media. If you need serum-free before you add your compound, you can put it back on the magnet, remove your media, and add your serum-free media. If you want to do the immunohistochemistry, same thing. If you want to do automation to do your washes, you can do that too. And we have users using automated washers to wash their cells and not losing their cells because they're being held magnetically on the bottom.
Can I perform biological experiments such as immunohistochemistry, Western blotting, qPCR, biochemical assays on 3D structures with nanoshuttle? Yes, you can do all the above. One thing that I tell people is if you have a protocol for monolayer culture, that's not necessarily the way to think about 3D culture. If you have a protocol for in vivo tissue, that's probably closer to how you should handle your 3D cultures. Because, for example, if you're going to lyse your cells, it's going to be a bit harder to lyse the cells in 3D than in monolayer. So you may need to be a little bit more forceful or use a little bit more stringent conditions. But, or even if you want to extract more DNA or RNA from your cells, you may need more cells just because of the nature of the 3D cultures. And that will also depend from cell type to cell type, because some cells make very tight structures that you got to work to break them up. But this is just inherited because we're working with cells assembled into a tissue configuration, not as a monolayer. Next question. The best possible scenario for imaging usually is when the samples are on the cover slip. After removing the magnet, do the spheroids tend to settle to the bottom, or do they stay floating?
They usually stay centered in place on the bottom. Yes. They are not attached, so in some ways they are floating, but they're not floating around, let's put it this way. So you can use the magnet because we use cell repellent surfaces and no gel, no matrix. So the cells are free on the surface, but the magnetic field can bring them to the center and hold them down.
We have so many great questions coming in, but we only have time for one more. Dr. Souza, what type of throughput is achievable? Realistically, how fast can you get through 1536 wells in multiple channels with adding the extra dimension of scanning through Z?
So if you let's take for example a 9.6 well, and then we'll go to I'll do a quick math for 1536. 9.6 well using 2 colors, 1 field of view. That will take about 2 minutes to go through the 9.6 well. So if you extrapolate that to 1536, it's about 32 minutes, I think, if my math is right, on getting a confocal stack on 1536 plate.
Thank you again, Dr. Glauco. Do you have any final comments for our audience?
No, thank you very much. If you have any questions, you can go to our website, n3dbio.com, and go to info and just contact us, and I'll be glad to address any of your questions. And we also, Grinder Bio 1 offers demos with our system too, if you're interested.
Thank you, Dr. Glauco Souza, so much. Now, audience, if you submitted a question through the Q&A button and it was not answered, someone will get back to you regarding your inquiry. Today's webcast will be available for on-demand viewing through March 29th of 2017. You will receive an email from LabRoots alerting you when this webcast will be available for replay. We invite you to forward that announcement to your colleagues who may have missed today's live event. Thank you again for joining us. See you next time. Goodbye.